Hello everyone, my name is Yasin Guryani, and in this video today I'm going to explain how mitosis and meiosis work. I'll walk you through each step of the process using 8 chromosomes for mitosis and 14 chromosomes for meiosis. I'll also explain which of these processes is involved in cancer development. Let's get to it. Let's start with mitosis. Mitosis is a process cells use to divide and create two identical copies of themselves. It happens in body cells like skin or muscle. Here are the main steps of mitosis. Step number 1 for mitosis is interphase. This is the phase before the cell division actually all begins. The DNA in the nucleus is copied, so our 8 chromosomes are now duplicated into 8 X-shaped chromosomes, each made out of 2 sister chromatids. The cell is now getting ready to divide. Step 2 is prophase. In prophase, the chromosomes become visible and the nuclear membrane starts to disappear. Spindle fibers begin to form from the centrosomes, which will help them move the chromosomes. Step number 3 is metaphase. In metaphase, the chromosomes line up in the middle of the cell along an imaginary line called the metaphase plate. Spindle fibers attach to the center of each chromosome. Step number 4 now is anaphase. During anaphase, the spindle fibers pull the sister chromatids apart. They move to opposite ends of the cell. This ensures new cell will get the same number of chromosomes. Step number 5 is telophase. In telophase, 2 nuclear membranes start to form around the chromosomes at each end. The chromosomes begin to uncoil and the cell prepares to finish its dividing. Step number 6 is cytokinesis. Finally, in cytokinesis, the cell's cytoplasm splits right in half, and we have now 2 identical daughter cells, each with 8 chromosomes. Alright, now let's move on to meiosis. Meiosis is the process that creates sex cells, sperm and egg, and it results in 4 unique cells with half the number of chromosomes. Since we're starting with 14 chromosomes, each final cell will now have 7. Step number 1 for meiosis is interphase. Like mitosis, it starts with interphase. The DNA is copied, so our 14 chromosomes become 14 X-shaped chromosomes ready to divide. Step number 2 is prophase 1. In prophase 1, homologous chromosomes pair up, one from each parent, and crossing over occurs. This means they swap parts of their DNA, which creates genetic diversity. Step number 3 for meiosis is metaphase. In metaphase, the chromosomes pair up. In metaphase, the chromosomes pair up, the line up right in the middle of the cell, and spindle fibers attached to them. In step number 4, anaphase, the pairs are pulled apart. Whole chromosomes move to opposite sides of the cell, not chromatids. Step number 5 is telophase and cytokinesis. This is where now 2 new cells will form, each with 7 X-shaped chromosomes. These cells are not identical, and they still need to divide one more time. Step number 6 is prophase 2. In prophase 2, spindle fibers form again in each cell. The chromosomes are still X-shaped, no DNA is copied again at this point. Step number 7 is metaphase 2. Chromosomes line up in the middle of each of 2 of the cells. Step number 8 is anaphase 2. This time, chromatids are pulled apart to opposite ends, just like mitosis. Number 9, telophase 2 and cytokinesis. Now 4 cells are formed, each with 7 single chromosomes. These are either sperm or egg cells. They are all different, which is important for genetic variation. Before I finish, let's talk about cancer. The process generally involved in cancer is mitosis. This is why it's important, and not meiosis. Cancer is caused by uncontrolled cell division, and since mitosis is responsible for making new cells in the body, if it happens too often or goes wrong, it can lead to tumor growth. Thank you guys for watching. I hope this video helped you guys understand the difference between mitosis and meiosis and how both of them work. Goodbye. Thank you.